Sri Gurubhyo Namaha. Welcome to your online learning initiative, and I welcome you all for the biology class. Last class we saw about incomplete dominance, codominance, as well as pleiotrophy and polygenic inheritance. In gene interaction, we are going to see the last part. It's nothing but epistasis. We'll see. So, what is epistasis? In the introductory class, I told you it is nothing but standing over, epistasis, standing over, not allowing the other character to express. Okay, that is called epistasis. Which is not expressed, you call it as hypostatic, hypo, below, right? So, what is the difference? Dominance also will not allow the other character to express, and epistasis also will not allow the other character to express. That is intra-allelic. Okay, this is interallelic. Okay, so I'll write here what it is. This is inter, between two. Inter, okay, interallelic, between two different alleles. That is epistasis. There, intra, between the same. If you are going to say tallness, capital T, instead capital T, small t when it comes. I'll write here. So to be more clear for you, capital T, small t. Between these two allele itself, this will be dominant, will not allow the dwarf character to express. This is inter or intra. It is intra-allelic. Okay, not this one, allelic, right? Okay, whereas interallelic, I can say capital Y. Okay, example, yellow color I'm taking, another one, white color I'm going to take. Okay, so between these two, interallelic, okay, between two different alleles, okay, and not allow the other. For example, this white is a hypostatic, not allow this character to express. It shows only this character, yellow color. Okay, so it is called epistasis. Now we'll still more explore this. Epistasis is the interaction between the genes that influences the phenotype, the character. I'm talking about your color. They can affect one another in such a way. So one another they are going to affect in such a way that regardless of the allele of one gene, okay, it is recessive to one dominant allele of the other. So here this character will be expressed, it is dominant. The other one simply it will mask, it will cover, not allow. Okay. An alternative expression to epistasis involve, we can show by ratio as well as chart. Okay. So I'll show you one example. Okay. Example we'll see next slide. Now I'll give you still more a detailed explanation. So here is the chromosome. Okay. We'll draw in pair. Okay. And then another chromosome, another set of chromosome. Okay. And now the gene we are going to write here. So here is the gene. Okay, capital Y, the same letter I will take it. And here the other gene. W, example. I'm just taking example. Small w, small w. So, it's an interaction between two different alleles, interallelic. And this will be dominant. It just masks or cover the other character and not allowed to express. So this is called as epistasis. Okay. And then the same thing in previous slide, I showed here intraallelic. Okay. That also I will show here. Okay. See. Yeah, chromosome. Okay. And the letter capital T, small t. Okay. It is inter or intra. Right here, it is intra. Okay, in the same homologous chromosome, interaction between the two genes of the same chromosome, it is intra. And this when two different non-homologous chromosome, it is inter. Okay, interallelic, right? So we'll see an example here. Before going to the example, some differences, okay, between the dominance and epistasis, as we discussed. So it involves intraallelic and it involves interallelic. Okay, have a view. Intraallelic, interallelic, right? Then one allele hides the effect of the other allele in the same gene pair. Example, we are using capital T, small t, same gene pair. One gene hides the effect of other gene at different gene loci. Okay, so Y, okay, then W, like that we can compare, right? Next, hypostatic versus epistatic. So what it is? Epistatic gene is a gene that can suppress or mask the expression of another gene. That's what we discussed. Whereas here, it's a gene whose expression is affected. Expression is affected, not allowed to express. Example, we are going to see dog's skin color, that is fawn color, that is coat color, we can say. Okay, Labrador dog, if you take it, fawn color will be there, then black color will be there. Okay, chocolate brown color will be there. That we are going to see as an example here. So here, it's just an example. A dihybrid cross showing recessive epistasis. We are talking here about recessive epistasis. Here, there is a condition. Even a recessive character can show their presence in presence of other character. Usually, the other character will be dominant, right? And not allow the other, the recessive character to express. Here, okay, a recessive, for example, here, which is recessive? The yellow color you can see. Even the recessive character can also be expressed, can become into a epistatic gene. So the two colors you can take black color and then yellow color. Okay. When it is intercrossed, you're getting a new combination brown color. Okay. Here also you will get a blending characters. So its yellow is nothing but this fawn color. It's a yellow color. So next topic, we are going to go into sex determination. Okay, sex determination mechanism, if you take homogamity, heterogamity, two types are there. Homo, when you say, it is nothing but the same. The well known, we will take it, human chromosome. So this is XX and this is XY. Which is homo and which is hetero? This is homo, same chromosome. This is hetero. Okay, based on the hetero only, the sex of the child will be expressed. Okay, so suppose if this Y chromosome in sex determination in human, we are going to see, okay, how this Y chromosome will have an influence over determining the sex of an individual. Okay, so I'll introduce here the type of mechanism. I'll erase this so it will be clear for you. Yeah. Finalization of the sex at the time of zygote formation. Okay, zygote formation, one chromosome from the father, another chromosome from the mother. Okay, they fuse and that will form as the diploid, right? That you call it a zygote. Formation is called sex determination. So this mechanism, if you're going to see heterogamity, I've classified here male heterogamity, another one female heterogamity. Okay, male heterogamity, human male heterogamity. What about the other organisms? That we'll see here. XXXY type, it's human. Another one XXXO type, grasshopper. The other two types we are going to see, something new we are going to see here. ZZZW, female. The ZW we are saying, right? It is a female. Okay, I'll write here. it is a female. And then ZZZO, here this is also a female. So as I said, here female or heterogamous, whereas here male or heterogamous, right? Okay, so we'll see what is that example. First, XXXY type. Okay, male have X chromosome and Y chromosome. Okay, so who is determining the sex of an individual? Here, the chromosomes are determining. In that, who plays a major role depending upon the Y chromosome. Heterogametic condition only we can say whether it is a male or female. All the homozygous in this XXXY type are female. Heterozygous XY type or male, right? So here, this is a male. Okay. The next, XXXO type. Here also, what is that O? O already I said in ABO blood group, O indicates absence. Here also O, absence of chromosome. So here, one chromosome will be less in number. That is nothing but male. Male heterogamety. Female homozygous. And next, ZZZW. Here, this is female. Okay, be careful while writing. This is female. It is seen in birds, fowls, and fishes. Okay, one Z and one W chromosome will be there in a heterogametic condition, that is female. The other two, both are same, ZZ, that will be homozygous male. Then ZOZZ type. It is seen in cockroaches. And here also one is absent. Okay, that is why the place is called as O, nil, 0. Right? Now the question corner. You can see the question. How is the sex determination mechanism different in the birds? In birds, what mechanism it is? See here. It is ZZZW. Okay. Now observe the question. Is the sperm or the egg responsible for the sex of the chick? Who is responsible? Egg will be responsible because it is having a heterogametic condition. The same question, if I put like this, human. In human, who is responsible for the sex determination of a child? It is male because the sperm only will carry either X chromosome or Y chromosome, right? In human, okay, from 8th standard we are learning about the sex determination in human being. Okay, just observe the chart. Okay, here 50% chances of male and female. Okay, female how we are denoting XX and male how we are denoting XY. Okay, pull out the gametes. Okay, and then as we did in monohybrid cross, intercross, you will get a ratio 1:1. Okay, percentage 50% is to 50% male chances or female. Okay, I mean 50% chances for male, another 50% chances for female, right? So who is determining the sex of the individual? The sperm is determining because it carries either X chromosome or Y chromosome. Why is it so? Because it is a heterogametic condition. In honeybee is quite different. Okay, if you see, there is a new concept called parthenogenesis. We'll see that. So here, okay, you can see the group of honeybee. I'll write the term, a new term you're going to learn, parthenogenesis. Okay, otherwise called as haplodiploid condition. Okay, haplodiploid. Female will be 2n, diploid. Male will be n, haploid condition. The male bee are called as drone. You can see here, drone. It's male. And the queen bee, female. Okay. If you see, this is a condition. All the male will be haploid and all the female will be diploid. That female, well-nourished female, will be the queen. And the remaining female are sterile. They will not give birth. Okay. So this queen bee will have two XX chromosome. It is a diploid condition. Whereas drawn male will have only one X chromosome. It is haploid condition. Again, the gametes are pulled out. Okay, here only one. So one gamete only can be pulled. Okay. And see here, this X chromosome, it is just egg. From the egg, due to mitotic cell division, they keep on divide. Okay. And it will become fertile male. Note down the term fertile. So they can mate with the female honeybee. Another one, this X chromosome from the queen and one other X chromosome from the drone, there is male as well as female, it will become into XX chromosome, and this will be sterile or they are fertile, depends upon the nourishment. Only one out of that female will be well nourished and they develop into a queen bee. Remaining all will become the worker bee and they are sterile. Okay, we'll see the flow chart still more. Okay, here it is nothing but the explanation about it. Note on this term alone, monoploid. Okay, only one. Yes, and that is nothing but drawn. So here is a flow chart. Okay, this flow chart very important. Okay, you may get 3 mark type question. Last year board question was from this only. Okay, female queen diploid condition haploid. Just now we discussed it is 16 number chromosome. Here 32 number chromosome. Okay, it undergo a division called meiosis. It undergo a cell division called mitosis. Okay, that is why there is a double the number. And here it is, they divide into, okay, in meiosis, how much you will get of the number of chromosome. It's a reductional division. In mitosis, how much you will get the same number of chromosome, equational division, right? So here I told you the term, right? Parthenogenesis. Okay, it is nothing but the development of the egg without fertilization is called parthenogenesis. Okay, I'll write here, this is without, the egg will develop without fertilization. Okay, so we'll get haploid male here. And here, after fertilization, they develop, that is diploid male. And the diploid male will have two category. Well nourished will be the queen bee. Okay, not well nourished will be the worker bee. But both are what? Female, right? So here is a question corner. Okay, a haploid, haplodiploid. Okay, not haploid, haplodiploid sex determination system as a special characteristic features. Special characteristic features such as the male produces sperms by mitosis. Okay, it is equational division. They do not have father and thus cannot have sons. Okay, it is like justification type question. But have a grandfather, can have a grandson. Okay, you are going to check this one and we will see the answer in next class. Right? Thank you.